going to do 2 stains. You're going to do the acid-fast stain, and we're using a particular acid-fast stain procedure, a particular method, the Zielen-Neethen method. You do need to know this, right? Because it determines what color our cells are going to be. Not so much for the carbofusion, right? No matter what method, they're always going to be that purplish pink color, the carbofusion color. But the counter stain tends to vary depending on which method you're using. Even within Zielen-Neethen methods, some people use brilliant green instead of methylene blue, but we're going to use methylene blue. We get pretty good results with it. Same thing with the endospore stain. There's a bunch of different methods out there. I like the method we use. The other method we used to do is not fun. You used to actually boil them in the test tube with the first dye and then take them out and make a smear. It was not fun procedure. This one, believe it or not, is easier than what we used to do. And this is the Schaefer-Fulton method, where we're going to stain a bacterial smear. Again, we're going to employ heat for both of these stains today. And there's a reason why we're going to use heat on our steaming apparatuses. So acid-fast staining using the Zielen-Neethen method. If you have got acid-fast organisms, right, they're going to be this purplish pink color. They're going to be bacillus shape. And because of the makeup of their cell wall, they tend to clump together. They're hydrophobic, right? So the water component of the dyes that we use really cause these guys to squish together. And of course the water that we use sometimes to even make the bacterial smear, right, will cause them to clump together. So they're rod-shaped bacteria, right, that retain the primary fuchsia-colored stains. And as I said, they clump together because they're hydrophobic. They fear water. Mycolic acid is a substance that they have associated with their peptidoglycan layer. So in addition to the peptidoglycan layer, they have this mycolic acid and the sugars associated with it. And so this is another extra layer of protection. So these guys are much more protected than gram-negative cells even, having this additional layer. And this additional layer is a waxy substance. So it's why we have to use acid alcohol as a decolorizer. And this is why it's called acid fast. So despite using acid and alcohol, hydrochloric acid, that's a really strong acid, it's acid you produce in your stomach, we do not normally wash away the carbofusion from the acid fast organisms. Fast meaning it stays put. Not as in I run fast, right? But fast as in it stays put. It stays adhered. Right? So despite acid, it stays there. That's where the acid fast stain gets its name. So because this is a waxy substance too, though, when we heat it, we kind of melt it, right? So like when you heat candles, right, and you have candle wax, anything that falls into that wax and then after it cools, forget about getting it back out, right? Ain't happening. Right? So that's what we're going to do. We're going to basically, well, melt the wax. The dye is going to get stuck in it. When the cells get cooled, it really sticks in those cells, right? That carbofusion. If they've got this mycolic acid, if they are acid fast, right, that dye is going to really stick in their cell wall. So normal phospholipids have about 10 to 20 carbons. Mycolic acids, the long ones, are about 50 to 60, and the shorter ones are around 20 to 26. That's a lot of basically fat. So pathogenic organisms, so one of the main reasons, right, why a scientist would stain and with the acid-fast stain is they're looking for mycobacteria. So they're looking for Mycobacteria leprae, right, which is, I did research in that, so there's some cool information here about that. In Peter's opinion, can't remember what that is, but I'm not going to test you on it, right? This is bonus stuff. Okay, the other that we're probably aware of is Mycobacteria tuberculosis, right, which causes tuberculosis. So this is very widely used stain, right, in those two research fields and for diagnostic in helping identify individuals that have these types of infections. So, and then there's one more, and so this is an opportunistic pathogen that can cause an infection of the heart and even affecting lungs and other body parts. Generally, it requires some degree of immune suppression, right? The immune system is not taking care of this invader. And it's transmitted inhalation. Peter didn't put it in here. I can't remember if this is bacterial or not, or if it's some type of other pathogen. He doesn't have it in here. But this one stains acid-fast positive, although not very common. This one I know, for instance, is a eukaryotic parasite that they also sometimes will use the acid-fast staining to identify. This isn't an acid-fast picture, right? But it shows you that the spores produced by this organism do get stained with the acid-fast and help us be able to see it. And so it's in the same group as Plasmodium that causes malaria, right? So similar type organisms for that. So those are mentioned in your book. By no means you guys need to memorize them, and just let you know that they're out there. Especially you guys going into allied health fields, right? Health fields. It's good to know, right, why we do the things we do, right, in lab. So you'll see that we already have our apparatuses set up, and I meant to tell you guys to turn them on before I started this lecture, and I forgot. Turn them all the way up. Crank them. And we probably won't need that one because I need to put that at the first slide of this lecture. Turn on hot plates. You got it? Yeah, all the way 10. Turn it all the way up. You don't want to, there's two plugs. You don't want to unplug your light box, though. Oh, yeah, Shawanda's not here, so yeah, you're good. And Byron Nisha left. We don't necessarily need that one. I could. Yeah, I'll leave it. Okay. So once those get steaming, what you're going to do, and as you can see, if you place them like this, if there are 4 people at your lab bench, which isn't even in the case today, we can fit 4 slides on it all at once. And I might ask you guys to stain an extra slide for me anyways, so we might do 4. So these I've already pre-made for you guys, again, like I did the gram stain. So I have an acid fast and a non-acid fast in 2 different circles. It's already heat fixed. It's ready. As soon as our hot pots are ready to go, we'll put those slides on. We'll put a piece of this filter paper that you guys have picked up, right, on the slide, and then you can put the carbofusion dye. You got to be wearing your goggles because stuff could splatter for sure, right, when you're over the watch pot. You got to keep this moist. You don't want it to dry out, right? So you want the whole time we're heating and melting that waxy substance of the mycolic acid of the acid-fast bacteria, you want it keep getting that dye in there, okay? You don't want it running over, right, everything and into the water per se, but you want this to stay moist. So someone has to babysit it, right? Someone in your group at one time, you can rotate in and out, but someone must babysit, right, the whole time for the 5 minutes that you're staining. Once you're done, and we'll talk about that in a moment, I'm going to show you the best way to remove them. So some questions as it relates to this staining procedure. How does the heat help us? Why are we heating? To melt the waxy substance, right, the mycolic acid. Right? Help liquefy it, help get that dye in it. The acid-fast negative cells are going to get stained with the carbofusion. Just like gram-negative get stained with crystal violet. The difference is when we decolorize, right, we're going to be removing the color from the ones that don't have that waxy substance, that aren't mycobacteria. So are acid-fast negative cells stained by the carbofusion? Yes. But what makes this procedure differential? We're going to decolorize. And this is important today. What is the decolorizer you must use? Acid alcohol. Make sure you grab that and not the plain old ethanol. Now this you're instructed to do dropwise, and in this case you want to pretty much do it until it's mostly clear. Most of the color has been washed off your slides. Don't do it too long though, right? But this is one of those ones where it's not as touchy as the gram stain for decolorization. Why do you suppose the acid-fast stain is not as widely used as the gram stain? So, who did we say is acid-fast? Only mycobacteria. That's one genus of bacteria, y'all. But with gram staining, it's going to be one or the other. And remember, how many did I say are gram positive? It's actually the one that's 40%. Most are gram negative. But pretty much half and half, right? It divides our possibilities in half. So the gram stain really gives us more information. Where acid-fast, most of the organisms that you're going to stain are going to be non-acid-fast, right? As we did when we were reviewing. All our organisms in the lab are non-acid-fast except for one. Who is acid-fast? The mycobacteria. So, and then in this one, look at, they even have malachite green as the counter stain. But we're going to use what? We're going to use methylene blue. So acid-fast are going to stay stained in the carbofusion, right? Even after acid decolorization. We will decolorize. These guys get really damaged with that really strong decolorizer. So yeah, the colors are kind of muted and not so pretty, right? We're not going to see beautifully blue cells, but they'll be bluish. We'll be able to at least see them instead of being colorless after decolorization and not be able to visualize them. So you'll have both on your slides. Of course, right, the mycobacteria, which is not this one, it's Smegmatis, is going to be, and see how it's really clumped in this one picture? And again, notice it's that pinkish purple color in all these pictures. Right? And if you see individual cells, they're rod-shaped. The non-acid fast we have on our slides are the Staphylococcus epididymis. So they're going to be cocci and clusters, and again, they're going to be kind of bluish in color, like you see in this picture and this picture. So, right, we can apply the scientific method to all the protocols that we do, right? There's a reason why we're doing it. We want to know if it's mycobacteria. Right, we have a protocol we need to follow. We're going to get results. We're going to look at them under the microscope, and those results are going to tell us something about that organism. For the endospore staining, right, endospores are a dormant form of the bacteria. This is not a form of reproduction. Unlike most other names, like when we say spore, we're talking about reproduction. Not the case when it comes to bacteria. Of course, at one time they probably thought it was a form of reproduction, hence why it got this name. But it is not. This is when conditions have become harsh. It's going to protect its DNA, the most precious thing inside that cell. So that when the conditions become good again, it turns back into that original cell it once was. So again, the terminology a lot of times sounds like reproduction, germination, going from the spore back to the cell. Sporalization, forming the spore from the mother cell. Again, mother cell. Sporangium. You hear these reproductive type terms when referring to this process, but it is in fact not reproduction. Just dormancy. Saprophytes, a lot of saprophytes are heterotrophic, which means they digest dead or organic matter. There are decomposers. A lot of these guys are found in the soil, right, that help decompose dead plants and animals. Heterotrophic, remember, means that they're using carbon in an organic form. So it came from an organism. Something died, right? We're saprophytes. We don't eat a cow while it's still alive, right? Plants, you know, there's, I don't know how you determine if it's alive or dead, though. Oh, it's kind of a tricky subject as far as it goes. I don't know how you determine that. But for the most part, right, we're eating stuff that's dead, right? We're saprophytes. We're heterotrophic as well, right? We're eating organic sources of carbon. Keratin is one of the major proteins in that spore coat. It's the same protein that makes up your hair and your nails and the outer layer of your skin, right? Really hard protein. And that's one of the main reasons why we've got to use heat today, again, for the endospore stain. Because this is going to help. Huh? No, it's a protein in your fingernails. And that's what makes them hard. Right? It's in your hair, it's in the outer layers of your skin in high concentrations. The highest concentration is in your nails, right? That's why they're so hard, because of that particular protein. So the same thing with the endospores. They have keratin, and to weaken that, we heat them, right? I have actually really strong, at least I did at one time, really strong nails. So much so that I cut them after I take a hot shower, because that's when they're softer, right? The heat and the water soften that protein and makes them softer and easier to cut, right? I unfortunately get ingrown toenails thanks to my dad. And so literally after I soak my feet, then we, try and cut the damn nail out of my toe. But again, the same thing here, right? We're going to heat them with moist heat, right, with steam, and that's going to make that protein more malleable and help really get the dye into the endospore so that we can see them. Otherwise, they don't stain, right? So, again, in the horns of this animal, that's keratin. That's what makes it so hard. Bacillus Anthracis, right? It's an endospore former. We're obviously not using in this lab because it's extremely pathogenic, right? But this is what went through the mail at one time. That powder was the endospores. And then when someone inhales it, it germinates, forms that bacteria, and unfortunately that's extremely damaging bacteria. It can kill you. And it is not found predominantly in the environment here in the United States because we've done a really good job of cleaning it up. Other parts of the world, not the case. Yeah, but we've done a really good job of making sure that animals that have died of anthrax, that we properly dispose of them so they don't become part of the soil in the United States. Not the case in other countries. Unlike tetanus, right? The cleanup effort for that one would be astronomical. But what do we do to protect ourselves instead against tetanus? Vaccination. And again, for this one, it's against the toxin it produces, not really the bacteria. You can still get the infection. Antibiotics will clear that up. It's a gram-positive bacteria, right? Good old penicillin will take care of it. It's the toxin that can really be lethal, right? And that's why we vaccinate against the toxin it produces. And again, it's a spore former. It's found predominantly in the soil. You can't avoid this, baby. It's everywhere. And then same thing with botulism. It can contaminate our foods. It's a toxin producer, and it's the toxin that hurts us. It is what they're giving you, that toxin, when they give you Botox, right? So, Clostridium perfringens is another one, a spore former found in the environment. And it can grow under anaerobic conditions, can cause really serious infections, commonly referred to as gas gangrene, because it actually produces gases and it'll cause the pus and stuff to bubble from the gas production as it's eating away at your flesh. Not a pretty one. But again, wounds that aren't properly taken care of and get this spore into it, this organism could germinate and grow and then cause really bad damage. So much so that, to the point that you would have to lose a limb or lose your life, right? Always, really serious wounds, make sure you get proper wound care. Clostridium difficile is one you may run into in hospital settings. This is another spore former, another one in the environment. Usually people don't get this type of infection of their intestines unless their normal bacteria are not there or have been compromised from overuse of antibiotics or other issues. This one will take up residency, and another one will eat you inside out. The other bad thing is another gas producer. So the people that have this type of infection, apparently horrific gas. Like, the patient has this infection before you even walk into their room. It's that bad. I haven't personally experienced it, but I've heard from others. Yeah, this one. You never forget this one. Right? So we've already gone over locations, right? Central, right smack in the middle. Terminal, right at the end. And then subterminal between the middle and the end. Sometimes the spore itself, you can tell that it's elliptical, right? Where others are more spherical, ball-shaped. And then sometimes it's even larger than the cell, so you get this swollen section where the spore is forming. One of the Clostridia looks like this. Can't remember which one. So vegetative cells just go through the normal cycle, binary fission, making more babies, right? All the same, actively metabolizing. A spore mother cell is where the spore is beginning to form, right? It's protecting the DNA. Eventually, over time, the entire spore mother will deteriorate, and then you have just spores by themselves. So depending on what phase our cells are in, you might see just cells, and you may just see spores by themselves. If we're lucky, scan around on your slides, we may see a spore still inside the mother cell. Right, so actively metabolizing, or we're producing a spore. We're saving our cells. Again, it'll come back out, right, and form the original cell, can go through the vegetative cycle under good conditions. So we're going to stain with malachite green. This is going to stain everything. The decolorization step is just water for this, but lots of water, right? So watch your trays. Don't let them get too full so you're not spilling it all over before you're dumping it in our funnels. And then we're going to counter stain with safranin so that we stain the cell and any cells that don't have a spore in them. And again, we may have just spores by themselves, right, outside of the cell. And they'll still be green. So as you see in this picture, most of the spores are outside the cells, right? And then you see the cells themselves in red. In this picture, you can see, right, spores by themselves and some inside the cells. And then some without spores yet. So remember, they're going to be greenish, right? Not going to be like beautiful, no St. Patrick green here, right? Not that bright, bright green we're used to. Okay, why does this exercise call for 5-day older or older cultures? Right, this is the harsh environment. What's the harsh environment? Nutrient depletion, right? We starve the babies. Right, so you should have spores to look at. And literally will happen within 48 hours for these organisms we're using. So we'll see spores today. The reason why I did that instead of 5 days is by 5 days we're mostly just looking at free spores, and we're not going to catch any inside the mother cell. What does a positive result tell you? Well, the organism. It's a spore former, right? If we don't see spores, which today is not going to be the case, right? Because the ones I gave you are spore formers. But what if I give you an unknown organism after break, and you stain it and you don't see any spores, does that mean your organism's not a spore former? No, it might be that it hasn't done it yet, right? It's not a true negative with spores, right? A true positive is definite, right? You're going to see the spores, you stain them, you're going to know that they're spores. So again, think about why, right? So again, our cultures are going to be old enough when you guys work with them, when you're doing your unknowns, that if it was a spore producer, you'll see spores. If it's not, you're not going to see them. So we don't really need a negative control, right? I don't need to give you a negative one. Why? If you know you did it right, you should at least see what? No. If they're not spore producers, right, you won't see any spores, but what will you see? You'll see the cells. If you don't see at least cells, then something went wrong. But the cells themselves act as our negative control, right? Does that make sense to you guys? Okay. All right. So spores don't stain easily, right? Hence the employing the heat because of the keratin. We got to make them soft. We got to get the dye in there. You've already seen spores, y'all. We used Bacillus cereus already. You simple stained it. You stained the cell, right? And then remember I said there was stuff outside that didn't really stain, but you kind of saw all these little things? Those were the endospores, right? Those were 7-day-old cultures. So again, there weren't any spores inside the cells, but there was a whole bunch outside of the cells. If you get lucky, right, depending on the age of the culture, again, we would see it inside, but it wouldn't be stained. But if you see something inside of a cell and it's not stained, does that mean it's an endospore? No, it could be something else. But it would give you a clue, maybe this is an endospore. So what would you do next? If you think you have an endospore, right, how would you confirm that? What are we going to do today to see endospores? You're going to stain them, right? So if you do a gram stain, right, and you see this thing inside your cell that isn't stained, or maybe outside, you're like, oh, maybe this is spores. So let's do a spore stain and see if it, in fact, is a spore. Because there are other things that could be there that aren't staining, other than endospores. If you want to know it's an endospore, you've got to do what? The endospore stain. You got to actually stain it. Okay, so scientific method. Then now my tips and tricks. So in order to use your time efficiently, I suggest, right, do the initial step of making your bacteria smear for your endospore stain. So what you guys are going to do, and this is another tip I didn't show you guys before, is take your slide and with your grease pencil draw yourself a circle. That's your target circle where you're going to put your organism. But flip the slide over so the circle's on the bottom, right? And then you're going to put your drop of water. Then you're going to put your bacteria in. Each table has a different bacillus, so make sure you write on your notes which one you got, which one your table is doing. Then after we make a bacterial smear, right, we do water, bacteria, what do we have to do? Yeah, so we got to put it on our light boxes to make sure it dries, and then we got to heat fix it before we can put it on our staining apparatus, right? So make those slides first. But you also can have somebody already babysitting because we're already steaming, because we jacked those babies all the way up. I'm going to give you your acid-fast slides for your table. Somebody can put the filter papers and stain them and be setting that up while the other people are making their smears and then switch out, right? So someone else babysit while that person makes their smear. Make sense? Okay. So we're going to do acid fast first, and then you guys will all do your endospores at the same time. Remember to heat fix. So after steaming, grab yourself for your table an extra staining tray. With your gloved hands, right? Instead of using these or anything else and sending stuff flying, your gloved hands is fine. Grab on either side of this screen. Take the whole screen, put it on that extra staining tray like you see here. Then remove your filter papers onto a paper towel and throw them in the trash. Okay? Then each person can grab their slide, put it on their tray, and finish the procedure. Do the decolorization and the counter staining for your procedures, right? So put them on a paper towel so we're not dripping dye everywhere, right? And then you're going to take your slide, put it on your tray, right? Rinse it with water, decolorize with acid alcohol if you're doing the acid fast stain, and then counter stain with safranin. Rinse, blot dry, then it's ready to look at. Endospore stain, right? We're going to put them all on here with filter paper, the same thing. Flood them with malachite green in this case. 5 minutes. Keep them moist the whole time. We're going to take a whole rack off, right? Divvy up each buddy's slides. Rinse with water. Counter stain with safranin. Counter stain for acid fast is what? Methylene blue. I think I said the wrong one a minute ago. I was thinking of endospore, right? So if we work together efficiently, we can get these done, look at the cool stuff under the microscope, and then of course I'll put my demos up over there as well for you guys to look at. Let's do it. Yes.